as you can see, the labs off to the side, these are the offline. So this is analytical chemistry. I don't know if it's kind of boring unless you're a chemist, but it's got gas chromatography, liquid chromatography, mass spec, and multiples of those. That's microbiology in that corner, so there are sterile transfer hoods and all the usual infrastructure you would associate with trying to deal with microbiology in controlled environments. And this is where you would take samples offline, gases, liquids, etc. There's centrifuges in that corner. Now, this is probably not, but that's what you're seeing as you walk by here. Everything on that wall practically is devoted to the nutrient management. And it's kind of bright here, so we won't stay very long, but that's because the cooling system is acting up. The acid and base, and then two different nutrient solutions, as you see here. This is straight out of the greenhouse, conventional greenhouse application of hydroponics, where you split up your concentrated nutrient solution, otherwise they will precipitate, and you have an acid and a base for pH control. So these just fall by gravity into the big mixing tank, and there's a pump there that's running continuously, circulates this and mixes it up, and pumps it inside. And I'll show you in this other chamber what happens in Northern, they're just doing some germinations in here under standard conditions, so we can, they're not doing any gas analysis or anything, so we can actually open this door. Normally it would be sealed with all three of these. Just need some butter on this one. This door weighs a ton, the chamber weighs about 7 tons. So it's very well balanced, I believe. So these are soybeans that have just been germinated. This is where the hydroponic system gets plugged in. You see that manifold up there? That's about the steady pace of nutrient delivery through that irrigation system. And it flows down, and these are little Rockwell cubes, which will never go into space. But and I'll show you what will eventually. But so this, and we have these little shields on here to keep the algae at bay. But the algae microbes win in the end. They just win. So that runs continuously. It feeds, drops down here by gravity. So there's a bit of a downhill slope. This drops by gravity, fills this big tank here, which in turn feeds the head of the pump. So it all just runs around in a big circle. And there's a bypass loop that keeps stirring up the tank, and then obviously water after the atmosphere gets saturated at whatever temperature you have, and at whatever pressure you're at now. Water vapor pressure is impervious to total atmospheric pressure. Water vapor is a component of atmospheric pressure, but it responds to temperature only. So you can change the pressure in here. Let's say you're at 100 kilopascals, polar Earth atmosphere, and you've got 2 kilopascals of water vapor at 25 degrees. My mental arithmetic doesn't allow me to work out what that relative humidity is, but it's reasonable, say 60 or so. So if you pull it down to 10 kilopascals, 10th of Earth's atmospheric pressure, in a very short time, 2 of those 10 kilopascals will re-equilibrate as water vapor if you don't change the temperature. So we can make any atmosphere in here we want down to a vacuum, and we can create a Martian environment, except for the temperature and radiation and gravity, and a few minor things like that. But we can create the same atmosphere composition. And if we wanted to, we could create the same ultraviolet conditions too. Ultraviolet is attenuated at about 200 nanometers, so that's UVC on Mars. Here on Earth, it's about 300 nanometers. So if you want a really instant suntan, you just lay out on Mars for a few minutes. You want to turn yourself a couple of times, you're good. Of course, skin cancer is shortly, quickly behind you.
So the reason for the strength of the container here is for pressure?
Yes.
Is there any plant that would grow at Mars pressure?
No.
No.
However, we don't have a problem at a tenth of Earth's atmospheric pressure so far.
Okay.
How long does soybean seeds take to germinate to that size?
Just a few days. Break a nail. There. So, why did somebody ask me about the pressure? What we're asking here is how low can we take the pressure and still mess up the atmosphere composition a bit, because obviously at a tenth of Earth's atmospheric pressure, if all of it was oxygen, you're still half the oxygen, or less than half the oxygen that's in ambient air, which is 21 kilopascals. So oxygen is the limitation. That limits how low you can go. And the reason we want to know how low you can go is the lower you can get the pressure, the less massive your greenhouse will be, and mass and energy is the currency of space travel. Remember, the money gets spent here on students and technicians and me, but the mass of the system. So virtually all of the research that we're doing here, and indeed all of the research that anybody anywhere on the planet is doing on biological life support, is directly or indirectly aimed at reducing the mass and energy cost of the food production system. Ideally, you'd like your greenhouse to be an inflatable structure so that you could roll it up, stuff it in a little mid-deck locker on a shuttle or a crew exploration vehicle, bring it up to the moon. In the end, probably deploy it under some sort of radiation shield. So you'll require some sort of supplementary lighting, ultimately, because the radiation shield will also shield out the free photosynthetic energy from the sun. But the cost of that free energy is probably genetic mutations in your plants. So one of the early questions is that okay? You know, for lettuce and stuff that you're not going to take from seed to seed, well, it is okay. You know, and use solar flare, or be subjected to a solar flare kind of radiation loads, and probably won't like it a whole lot, but it'll probably taste the same dish. Put enough salad dressing on it. And so those are the kinds of simple questions that we'll have to figure out. And then we'll advance slowly but surely beyond salad machines to full-scale staple production like potatoes, wheat, rice, soybeans, beets, carrots, you know, the usual gamut of nutritionally balanced food based on a nutritional diet that's already been set up. 10-day, say, menu cycle for a crew of 6. And you need about, at the moment, based on our mass and energy requirement, you need about 60 square meters of plant production area for each individual crew member to supply all the food and life support requirements that they need.
For how long?
Forever.
Forever.
Yeah. So part of it would be in a batch culture for soybeans and wheat. Part of it would be in sequential physiological stages in many salad crops, you know, just fresh stuff that you would just sow enough of and then eat it. In the meantime, you'd sow some more, and it would be staggered production. You'd also have to have different parts of your life support system under different photo periods, because if it's all in the same day, then your oxygen goes up and your CO2 goes down during the day, and just the reverse during the night. So you have a really sawtooth atmosphere management, because the capacitance is so small. It's such a small volume. You're trying to reduce your volume and keep the mass down, keep everything small and tight. In any case, they can do a lot of the same things. You notice those down here. These are the ones that don't tend to break as often for some reason. Plus, I think they're closer to the lab where the guys actually work on them. But these are, you'll recognize this as a mosquito screen. We call it a neutral density screen. And it's designed